Hello, everybody, and welcome to our continuing webinar series. Today, we are really thrilled to have Dr. Rachel Keith, who's a postdoctoral fellow at the NIH, in the lab of Dr. Michelle Antoine. So our company has known Dr. Antoine for a long time in her work in neuroscience. And now we have one of her mentees, Rachel Keith, who is a rising star at the NIH. So, Dr. Rachel Keith earned her Ph.D. in neuroscience at George Mason University in 2022. And her dissertation focused on determining the influence of NMDA receptors on spatial learning behavior, hippocampal plasticity, and dendritic morphology across the postnatal period, resulting in 3 first author publications. So, again, she is one of the rising stars in the lab. And currently, as a postdoc in Dr. Michelle Antoine's lab, researching the neurobiological underpinnings of spatial learning deficits in mouse models of autism, she is going to today present her findings in her work in the medial temporal lobe, as well as her work in learning about the key roles in spatial navigation impairment in autism mouse models. So with that, Dr. Keith, I want to hand it over to you and welcome you, and thank you very much.
Great, thank you. Okay, so I'm going to assume my audio is good. So hi everyone. Today I'll be presenting to you my findings about how the perirhinal cortex may be mediating some of these spatial learning deficits that we see in 3 distinct models of autism spectrum disorder. So to kind of provide a roadmap, I'll be going over a brief introduction to autism spectrum disorder and some genetic models of autism spectrum disorder. I'll also give an overview of the method that I used, as well as the multi-level analysis that I employed. I'll present the findings themselves, and then I'll go over the implications and the conclusions of the results. Furthermore, what everyone should be able to take away from today is you should be able to identify some neuroscience techniques to determine the biological bases of behavior. You should be able to understand how tissue sectioning can be used in many ways to support neuroscience research inquiries, recognize the translational potential of animal models in neuroscience, and know more about autism mouse models and their neural activity in general. So autism spectrum disorder is a lifelong neurodevelopmental disorder, and it is diagnosed based on a set of core diagnostic symptoms. Approximately 1 in 36 children in the United States are diagnosed with autism spectrum disorder, and this number is increasing pretty much every time the CDC does its measures, which I believe is every 4 years. So diagnostic symptoms of autism spectrum disorder include social and communication deficits, restricted and repetitive behaviors or interests, atypical responses to sensory stimuli, and delayed cognitive or learning skills / intellectual disability. And I've included this graphic here to kind of illustrate the various aspects of the autism spectrum and various differences in functioning that may be present in an individual who has ASD. So unfortunately, there are currently no pharmacological or neurological treatments that have been shown to improve any of these diagnostic symptoms. And while other components of ASD can often be accommodated for through more accessible resources, greater understanding of neurodiversity, awareness, acceptance, intellectual disability and delayed cognitive or learning skills remain a challenging, heterogeneous aspect of ASD. For example, often autistic people may struggle with things like executive functioning, spatial navigation, novelty recognition, and object placement recall, leading to them having reduced independence, a reduced quality of life, and it could often lead to self-esteem issues alongside increased risk for anxiety and depression. So this factor of ASD is really important to study. And so our research aims to investigate the neurobiological underpinnings of intellectual disability and/or learning dysfunction using distinct genetic models that may lead us to treatments that are generalizable to many ASD patients. So there are a lot of genetic contributions to ASD. There are over 100 plus de novo gene mutations associated with ASD, and among them, 3 are particularly strongly associated with ASD. SCN2A, which is a gene that codes for the alpha subunit of NaV1.2, which is a sodium channel important for action potential initiation and backpropagation. And I have included the protein ribbon model here. There's also FMR1, a gene that codes for FMRP, which is an RNA binding protein involved in synaptic transport, also the ribbon model here. And then CDKL5, which is a gene that codes for CDKL5 kinase, a protein involved in neuron migration and synaptic transmission. Now, while mouse models that reduce gene expression of these three genes can be used to investigate the neurobiological mechanisms of ASD, it is only because the disorders that they model are so associated with ASD. For, in particular, SCN2A haploinsufficient mice, which I'll be referring to as SCN2A het mice throughout the talk, are going to be modeling SCN2A disorders, whereas FMR1 knockout mice are going to be modeling Fragile X syndrome, and CDKL5 knockout mice are going to be modeling CDKL5 deficiency disorder. And again, all of these are highly associated with ASD. Fragile X, for example, is the most common single gene disorder associated with ASD. SCN2A loss of function mutations display a penetrance of ASD of up to 50%. And CDKL5 patients often also display ASD levels of up to 50%, especially because symptoms in those patients are so similar to Rett syndrome, which is a pervasive neurodevelopmental disorder with intellectual disability. So even though there's a lot of gene mutations associated with ASD, these three are particularly strongly associated with ASD, which is why we chose them for our models here. So one crucial aspect that we are trying to investigate here is this kind of cognitive component and the ability to learn. And learning and memory often form the basis of how we interact with our environment and respond to adversity or challenges in our environment. But the problem is that when we're working with animal models, we can't really give them an IQ test and we can't ask them specific questions such as, do you remember what you had for lunch yesterday? And so as a result, we have to determine how these animals are learning through a more indirect measure, rather than simply asking direct questions. And so there are, many different types of learning, but I've highlighted three here: motor learning, visual spatial learning, and declarative learning, which is kind of learning associated with facts and procedures. Also, similarly, memory has three distinct aspects that I've highlighted here: spatial memory, episodic memory, such as memories of what you had for lunch yesterday, or memories of your own life, and motor memory, such as what we commonly refer to as muscle memory. So we're looking for something that we can use to investigate cognitive function in rodent models that employs aspects of all of these facets of learning and memory. And so we use spatial navigation to do this, because spatial navigation relies on several aspects of all of these facets of learning and memory. And so it serves as a great proxy for evaluating general cognitive function. And so I've included this graphic here to show how we might understand the type of learning that might be going on in a mouse or a human that is impaired in their spatial navigation ability versus not impaired. So if you were to let a person or a mouse navigate a complex maze, such as, this cityscape displayed here, a direct route would be considered something like this, where they take one turn and they make it directly to their destination. Someone who is impaired, who has, been shown the destination and has gone there before, may nonetheless sort of show this meandering path and therefore be somewhat impaired in their spatial navigation ability. So as a result, we use SCN2A het, FMR1 knockout, and CDKL5 knockout mice as our ASD ID mouse models. And we're going to be assessing the neurological basis of spatial learning deficits in ASD using a multi-level approach from cellular to behavioral, which I will go over now. So specifically, the test that we use for assessing spatial navigation is the Barnes maze. And so here you can see that it is a round platform that is iterated with, I think in this particular case, it's about 20 holes. They are evenly spaced apart. And one of these holes will be deeper than the other holes, which will be shallow. And it will usually be attached to a container that will permit the mouse to escape into and hide in order to feel comfortable and safe. And so because it is often, on a high platform under bright lights, it incentivizes the mouse to find this escape hole and then hide in the compartment until the test is finished. So to go over kind of our paradigm that we use in our lab, we have 4 days of testing. On the first day, they are also given some habituation trials whereby they're allowed to explore the maze for 5 minutes and kind of get used to the maze. Then there are 4 3-minute trials per day that are 30 minutes apart for each mouse. And we score the maze based on whether or not the animal has employed a spatial approach or a serial approach. The spatial approach is defined as having fewer than 3 hole selection errors before entering the escape hole, as you can see here, where C is considered a correct choice and I is considered an incorrect choice. So if they make fewer than 3 incorrect choices, we consider that a spatial approach. By contrast, the serial approach is where they make greater than 3 errors before selecting the escape hole. So in this case, they often meander from quadrant to quadrant until they reach the escape hole, similar to the diagram I showed before. And we also use imaging in our project, and specifically we're going to be imaging 2 kind of types of imaging we'll use is light sheet imaging as well as fluorescence microscopy. And so the light sheet imaging that we have employed in our project is going to be looking at CFOS, which is a protein highly associated with kind of acting as a protein marker for neural activity. And so here in this image, we actually are seeing a horizontal brain section. And in this particular instance, this is a mouse that is a FOS-GFP from a FOS-GFP line, and this is going to be showing the CFOS positive cells in green here. And then similarly, we use fluorescence microscopy for many different applications, but one of them is going to be that we are going to look at Cre recombinase, which is an enzyme that allows us to have very tightly spatially controlled expression of particular genes of interest. And I'll go in more depth about how we're going to be using these Cre mouse lines later on in the talk. And then we also use some electrophysiology to assess the cellular function in discrete regions of the brain, as well as determine how neuroplasticity may be affected. And specifically, this trace here is what we might see as an example trace of the cell populations that we record from. The stimulus artifact has been edited out just because it's this huge downward or upward spike, and it's not very pretty. So in this case, this first initial descending phase of the trace is the fiber potential, followed by the secondary descending phase of the excitatory postsynaptic potential. And so I'll go again a little bit more in depth into what these mean and how we measure them later on in the talk. And then this image is just showing the stimulated electrode and recording electrode, in this case, in the hippocampus. The viral injections we do for this project are specifically to inject viruses of a particular set of types, where in the left here, we have a GFP Cre. So this is just, again, Cre recombinase that has been tagged with GFP. And so anywhere that you can see GFP is where the virus is expressing. And then over here on the right, we also have a CAM kinase 2 HM3DQ cherry, which is going to be an HM3DQ protein, which is a designer receptor endogenously activated by a designer drug. And this is specifically going to increase intracellular calcium in the cell and allow additional neural activation above baseline levels. And so this HM3DQ is coupled to the CAM kinase 2 promoter. So HM3DQ is only going to be expressed on excitatory cells. And then it also has an M-cherry tag, which is what allows it to show this red signal here. So we use these for kind of distinct applications in the project. Again, as the results come up, I'll go over them more in depth. But if people have questions at the end, I'm more than happy to go into them in more depth. So the first portion of the results is our SCN2A het mice, and we have replicated the previous finding in the literature that SCN2A het mice have spatial learning deficits. And so you can see here that in the gray, we have our wild type, which is denoted by our SCN2A++ here. And then the SCN2A het mice, by contrast, are in the darker bars here. As you can see by the statistical comparisons I've included, the wild types learn across the test days and show increased spatial approach trials. As you can see on the x-axis here is the % of spatial approach trials. And so the wild types are learning and they're improving in the Barnes maze task. By contrast, though, the SCN2A het mice do not improve over time, and they show significantly fewer spatial approach trials on day 3 and 4 compared to their wild type litter mates. So they do have this impairment for sure. And then when we used light sheet imaging to see how their neural activity looked, we used this pipeline here. And so essentially we're going to detect antibodies for CFOS, which is again, the protein associated with neural activity, as well as NU-N, which is just a marker for neurons to make sure we're only looking at neural activity and not say glial activity. We then have the light sheet imaging occur. We produce images, detect the cells. We do some algorithmic changes to the images to make sure they're high quality, that the algorithm is matched, that the cell counts are accurate. And then we have the atlas alignment to the Allen Brain Atlas to get cell counts and cell density counts for each region of the brain that the Allen Brain Atlas has identified. Here in the first set of images is our wild types from FOS EGFP mice. On the left, we have the FOS EGFP signal, and on the right, we have the NU-N signal. And in the top row, we have from the primary visual cortex, and in the lower row, we have the perirhinal cortex. And similarly, we have the same images for the SCN2A het mice here. And so when we looked at all of these different regions across the brain and we constructed a heat map to determine how active neurons were in our wild types versus our hets, we do see that the SCN2A het mice show this predominance of less cortical activity compared to the SCN2A wild types. And so you can see that some areas where the differences are the greatest are a lot of these sensory areas as well as medial temporal lobe areas. And so since a predominance of these areas are cortical, I believe 21 of the 31 regions are cortical, we sought to investigate the role of the cortex in mediating this spatial learning deficit. However, there's been a lot of association as well of the hippocampus with spatial learning, and yet, interestingly, the hippocampus did not appear in our assessment of CFOS. And so one of the things we sought to do was determine if the hippocampus played a role in mediating the spatial learning deficits seen in SCN2A het mice. So to do this, we used an EMX1 Cre mouse line, which expresses Cre recombinase widely across the cerebral cortex and also in portions of the hippocampus and nowhere else. And so as a result, we will only have loss of SCN2A in those areas of the brain. The thalamus and cerebellum and other regions are spared. They have wild type levels of SCN2A. And so when we characterize their behavior seen in this graph on the right, we see that the wild types shown in the gray bars here do, again, learn over time, whereas the EMX1 Cre SCN2A flox mice, which again are going to show that loss of SCN2A in these Cre recombined regions here, do not improve in their learning over time. And also by testing day 4 show a significantly reduced amount of spatial approach trials. And so again, that suggests that the areas that are implicated and shown here in the EMX1 Cre line are critical for this intact spatial learning behavior. So in order to determine whether it was more of a cortical attribute or a hippocampal attribute, we used the DRD3 Cre mouse line. It got a little cut off here, probably because I resized the text, but that's okay. So here we see that Cre is predominantly recombining in the hippocampus. It's very strong levels of Cre recombination there. It also has some sparse Cre recombination in some areas of cortex, predominantly in layer 2-3. And I'll go over a little bit later the expression patterns more discretely across the brain areas that we are looking at. But by contrast, compared to the EMX1 Cre line, the DRD3 Cre SCN2A flox line shows improved learning over time. And compared to the original SCN2A het population, the DRD3 Cre line shows significantly increased spatial approach trials on day 3 and day 4, suggesting that these mice do not recapitulate the spatial learning deficit that we see, unlike the EMX1 Cre line, which does show the spatial learning deficit as in the hets. So when we looked at the CFOS CFOS marker in these mice to determine neural activity, we did see that there were 16 regions that showed reduced neural activity in the EMX1 Cre SCN2A flox line. Specifically, these were all cortical regions. Again, the hippocampus was not implicated here, which tracks with our behavior results here. And so from this sort of heat map that we see, there's all of these regions that have reduced neural activity. we created a putative spatial learning circuit. And so this is going to show how the sensory cortices, areas of the medial temporal lobe, and association cortices are interconnecting. And while the hippocampus itself was not directly implicated, it does connect heavily within the medial temporal lobe and also has connections with some of these regions. And so I included it in the circuit diagram just to kind of add another dimension to this aspect. And so kind of the takeaway of the slide is that the loss of SCN2A in the hippocampus does not impair spatial learning. And so we're kind of then making the conclusion of, okay, it has to be, some aspect of the cortex. And so previous work in Dr. Bender's lab has looked at the role of LAIR5 pyramidal cells in mediating some of the effects of SCN2A het mice. And so we sought to see if there might be a LAIR5 specific effect by using the RBP4 Cre mouse line, which that is going to show Cre recombination in dentate gyrus and some areas of CA3, as well as very strong recombination in solely LAIR5 of cortex. And so when we run these animals in our Barnes maze, however, we do not see any spatial learning impairment. By testing day 2, 3, and 4, the SCN2A het mice show significantly reduced spatial approach trials compared to our RBP4 Cre line. So that suggests that LAIR5 is not going to be mediating all of the spatial learning deficit here. To kind of follow up on this, we also used a LAIR6 model, which is the NTSR1 Cre line. And as you can see here, it recombines very strongly in LAIR6, as well as some thalamic areas here. And so when we ran these mice in the behavior, we also do not see a spatial learning deficit. They show behavioral spatial approach trials across testing days that are similar to SCN2A flox littermates. So these are going to be essentially the wild type counterparts within this line. And so they show identical learning. It's not different, and it's not comparable to the SCN2A hets. So that suggests that it's definitely not a layer specific component, or if it is, it's, it's unknown. And so we were thinking a potential path to move forward is to look at the Cre recombination across these 4 mouse lines to determine the spatial distribution of how Cre is recombining in the EMX1 Cre, the DRD3 Cre, the RBP4 Cre, and the NTSR1 Cre. And by doing that, we can hopefully narrow down regions where SCN2A loss is very important. And so when we did this, we looked at all of the 16 regions that were implicated in the original EMX1 Cre dataset, and we looked at how Cre was recombining in all 4 of these Cre lines in each of these regions in layer 2, 3, layer 5, and layer 6, just to see if there's a layer specificity there. And we mapped this to the spatial learning outcomes that we saw, either intact or impaired. And so what we saw is that only the EMX1 Cre line shows Cre recombination in perirhinal area 36 in layer 2, 3 specifically. None of the other Cre lines showed recombination in this region. So it's possible that SCN2A is crucial in this region for mediating spatial learning deficits. And to kind of make it clear what this area looks like, I've included this little diagram of the rodent brain. And so this entire aspect here at the area 36 and 35 is the perirhinal. This POR area is the postrhinal, and then it has also some cortical areas around it, which again are very highly reciprocally connected with the perirhinal cortex. And so to kind of illustrate that once again, I've brought back in the previous circuit diagram, which showed that the perirhinal area, again, receives lots of input and reciprocal connections with not only all of the medial temporal lobe regions, but also with sensory cortices and association cortices. So this kind of makes it a really good candidate for being a spatial learning kind of, I guess for lack of a better term, like this key node in this circuit. And because it's highly interconnected and, has all of these reciprocal connections within the medial temporal lobe, when we looked at the literature, we did see that there was a lot of previously found studies that show that there is this spatial property of perirhinal, and specifically that the perirhinal cortex may be coordinating spatial information processing in other brain regions, including the hippocampus, which again might explain why, even though the hippocampus itself did not show reduced neural activity, why the hippocampus may still nonetheless potentially be playing a role here. So we wanted to examine, following this layer 2-3 perirhinal cortex. And specifically, we used electrophysiology to do this. We wanted to examine not only baseline synaptic transmission, but also long-term potentiation. And long-term potentiation is the cellular and molecular correlate of learning. Essentially, cells that fire together wire together. And so in various different types of learning behavior, such as spatial learning, you will often see long-term potentiation result. And so the most behaviorally salient kind of protocol for, or stimulus protocol for inducing long-term potentiation is called theta burst stimulation. And so that is the stimulation protocol that we used to induce LTP in this region, just to see how the SCN2A het mice might be showing changes in LTP. So here are the results. And so in this first trace, which is from our wild type mice, the lighter trace is going to be our baseline measure. And then the darker trace is going to be the potentiated trace following the theta burst stimulation after 60 minutes of recording. So in our SCN2A het mice, you can see that the lighter trace, the baseline there, and the darker trace are very similar. There is not much growth of the postsynaptic response, if any. And then we also wanted to determine if loss of SCN2A in the perirhinal alone would mediate any sort of similar long-term potentiation changes. And so when we look at the normalized excitatory postsynaptic potentiation amplitude, we see that following theta burst stimulation, while the wild types are kind of increasing their potentiation over time, and these results are summarized on the right, they are showing, again, significant increases in the postsynaptic response over time. Our SCN2A het mice and the SCN2A knockdown in perirhinal mice do not show any sort of increase in potentiation over time, and they are otherwise indistinguishable from each other. So that suggests that this impairment in long-term potentiation in the SCN2A het mice is not necessarily being mediated by SCN2A loss outside of the perirhinal cortex, since these two mouse lines show pretty much identical long-term potentiation. And so kind of to follow up on that, we wanted to see if loss of SCN2A in the perirhinal specifically was sufficient to impair spatial learning. And so this is going to be a kind of representative slice of our viral injections here. And so I have the cortical layers labeled as well as areas of hippocampus, and then the perirhinal cortex is labeled here with area 36 and area 35, both labeled, and the rhinal fissure, which is a landmark used to identify the location of the perirhinal. So you can see that, again, we have Cre recombination in the areas that we are expecting it bilaterally. And so when we compare these mice, the SCN2A knockdown mice, to our DRD3 Cre line, we do see that the knockdown mice show significantly reduced spatial approach trials by day 3 and 4. So that suggests that SCN2A in the perirhinal is crucial for intact spatial learning. And so what we wanted to see is also if we could take a previously learning mouse, the DRD3 Cre SCN2A flux group, which again shows intact learning and improves learning over time. We wanted to see, okay, if we induce the loss of SCN2A in the perirhinal in this model, which previously did not have this recombination, do we also see this impairment of spatial learning? And in these darker bars, yes, we do. We can see that they don't show increases in learning across time, at least not anything significant. And by day 3 and 4, they also show a significantly reduced amount of spatial approach trials compared to the DRD3 Cre SCN2A flux counterparts, and they are indistinguishable from the SCN2A knockdown in solely the perirhinal here. So that suggests that the perirhinal is in fact critical for having intact spatial learning behavior. And so if you remove SCN2A from this region, then you will have impaired spatial learning. So given this sort of notable impairment, we wanted to see, okay, theoretically, the loss of the alpha subunit of NaV1.2 is going to mediate a small, kind of a reduced amount of neural activity, which is confirmed in the CFOS results. And so we wanted to see, does increasing intracellular calcium via the HM3DQ DREAD potentially rescue this impairment? And so what we were able to do is we were able to have HM3DQ expression be mediated by the EMX1 promoter. So that would then mean that we have HM3DQ expressed in the cerebral cortex and hippocampus, same as previously mentioned. And so what we would do is we would take these mice and we would give them the endogenous, or exogenous, I should say, exogenous, chemical known as clozapine N oxide. And so we would give this at a dose of 0.25 milligrams per kilogram prior to the behavior test, about 30 minutes prior, to allow the drug to wash in. And so then we would run these in the same behavior tests that we've run the previous SCN2A wild types and SCN2A hets. And as you can see, compared to the hets, the mice which express HM3DQ in the cerebral cortex and hippocampus do show improved learning over time. And by day 3 and day 4, they show, or actually just by day 4, they show a complete rescue to wild type levels of the spatial approach trials in our Barnes maze. And so that suggests that, okay, increasing intracellular calcium can in fact rescue the spatial learning deficit. However, we then wanted to see if solely doing this increase of intracellular calcium in the perirhinal would be sufficient to also rescue the spatial learning deficit. And indeed, that's what we see. So in these orange bars again, we show that these mice are able to show an increase in spatial approach trials by day 4. From day 1 to day 4, they show this improvement in learning compared to the SCN2A het mice, which again do not show this increase in learning. And this is a relatively small sample size here. It's only 5 mice. Which I believe is why there's no kind of inter-test day significance. But nonetheless, we do see that there is this improvement in learning over time in the HM3DQ mice, which show, again, HM3DQ in solely the perirhinal. And so they do show this increase in learning compared to the hets. So then we wanted to see how our chemogenetic rescue and increase of intracellular calcium might be affecting long-term potentiation. And so we have these results here, where we again take the EMX1 HM3DQ flocks SCN2A het mice, and we assess their long-term potentiation in layer 2-3 of the perirhinal. And when we do this, following theta burst stimulation, we do see that our EMX1 HM3DQ flocks SCN2A het mice do show this restoration of long-term potentiation to wild type levels, which again is summarized here. And they show significantly greater EPSP amplitude at 30 minutes and 60 minutes compared to the SCN2A het mice, which again show this spatial learning impairment and this long-term potentiation impairment. So that's suggestive of the fact that long-term potentiation in the perirhinal may be the cellular and molecular mechanism that is driving intact or deficient spatial learning. And so while this was very nicely wrapped up in the SCN2A mice, where we could induce the spatial learning deficit and we could rescue it, we wanted to see if this rescue was applicable to other mouse models of autism spectrum disorder that have also been seen in the literature to show spatial learning deficits. And so, as I mentioned earlier in the talk, we looked at FMR1 knockout mice and CDKL5 knockout mice. So here we have an image of the perirhinal cortex, again showing the CAM kinase 2 HM3DQM cherry virus and its expression here. And we have the cortical layers as well as area 36 and area 35 of the perirhinal. And so in the CDKL5 knockout mice, in the spatial approach trials across time, they do not show an improvement in their learning. In fact, I would say that these mice were the most severely impaired in this task of all the 3 mouse lines that we tested. But by contrast, mice which were injected with the virus and given CNO prior to the behavior test do show an increase in learning over time. and by test day 3 and 4 show significantly more spatial approach trials than the CDKL5 knockout counterparts. And similarly, in the FMR1 mice, again, there's no improvement in learning over time. But when we have mice that are treated with CNO and were injected with CAM kinase 2 HM50QM cherry in the perirhinal, we do see that there is a increase in the learning ability over time from day 1 to day 4, and that by testing day 4, there is a significant increase in spatial approach trials compared to the FMR1 knockouts. So that is really, really compelling evidence that the perirhinal cortex is critical to spatial learning, not only in the SCN2A het mice, but also across different mouse models of autism spectrum disorder. This is really exciting because any mouse model of autism that has spatial learning deficits could then potentially be investigated for perirhinal cortex dysfunction. And so we could kind of use this as a springboard for future results. So to go into the conclusions of the findings, the perirhinal cortex is critical to intact spatial navigation in these mice. We have 4 lines of evidence for this. So the first is that, oh, I think these are out of order. Okay, yes, those were out of order. That's okay. So the first is that SCN2A knockdown in the perirhinal cortex alone is sufficient to disrupt spatial learning. And then additionally, SCN2A knockdown in the perirhinal of a previously learning mouse line also impaired spatial learning. However, loss of SCN2A in the hippocampus of the same mouse line did not impair spatial learning. So that suggests that even if the hippocampus is involved in the spatial learning impairment in the SCN2A het mice, which has been suggested by prior literature, it's likely downstream of the perirhinal cortex. Additionally, LTP is disrupted in the perirhinal cortex by SCN2A knockdown, specifically in the perirhinal, and then also in the EMX1 Cre line. And then lastly, increasing the neural activity of the perirhinal in 2 additional mouse models of autism spectrum disorder was sufficient to rescue spatial learning deficits. So some future directions that we have for our research are, we definitely intend to further investigate the perirhinal cortex to hippocampus circuitry, because there has been some research and literature out there showing that there are direct connections of the perirhinal to the hippocampus, whereas before it was mostly considered that the perirhinal served as a relay through the entorhinal to the hippocampus. So we're very interested in direct connections to see how they may be mediating each other's firing and to see if there may be a neural activity component. And then we would also like to investigate the perirhinal cortex in other domains of ASD, such as mood and sensory differences that are often present in these patients, to see if the perirhinal could also be playing a role there. There has been previous literature that's shown the perirhinal is involved in fear conditioning, as well as potentially aspects of visual processing. So we will be looking into that. Additionally, we do want to assess the perirhinal cortex neural activity levels in FMR1 and CDKL5 knockout mice to see if the perirhinal cortex itself is innately hypoactive at baseline, or if it is mostly mediated by a lack of, for example, evoked plasticity. And we'll be able to do this using in vivo fibrophotometry. And then we also want to translate these findings into more non-invasive methods, because obviously we can't use, intracerebral viral injections into human patients. And so instead, you can use things like transcranial magnetic stimulation and focused ultrasound to potentially improve symptoms in patients who are struggling with spatial navigation non-invasively. And then lastly, these mouse models often co-present with epilepsy, similar to patients that have SCN2A loss of function mutations, Fragile X syndrome, and CDKL5 deficiency disorder. And so we want to investigate a potential link between ASD-ID and seizure risk to see if there might be some interplay between those 2 conditions and if they might be related in a mechanistic way. So thank you all for listening to me today. This is the section on neural circuits and all of the people who have been involved in the research that I presented to you today. Dr. Michelle Antoine is my PI, and then I also have had the pleasure of working with Dr. Yiming Chen and Dr. Priscilla Correa Antonello in, they are fellow postdocs in working on this research. And then I also have had the pleasure of working with Jordan Jansen-Meza and Joey Abramovitz in this research, and they are former postdocs. They have since gone on to very prestigious graduate programs, and I congratulate them, and I hope they're doing well. And then we also had a summer intern who did a good amount of legwork on a lot of the imaging involved in this project, and she is currently working, Aisha Hamid is currently working on her master's degree. We've received a ton of grant support, and we also had the help of additional research assistants, Virginia, Oluwaseun, and Alani. So again, thank you all for listening to me today. It's been a pleasure to have this opportunity to present.
Wonderful. Well, it's our pleasure and honor to host you, Dr. Keith, in learning about the perirhinal cortex and long-term potentiation in autism mouse models. In our guest room, we actually have a couple of questions already. So I'm going to go ahead and moderate, and I hope I do these questions justice. Some of the questions were entered in the beginning when you were talking about mouse models and autism. The first one, there are 4 questions incoming. So the first one is for the de novo gene mutations you mentioned associated with autism spectrum disorder, how strongly are they associated? So for example, you mentioned SCN2A, FMR1, and CDKL5. Like what are kind of the statistical metrics for association?
That's a great question. So a lot of that comes from the Satterstrom et al. 2020, I believe is the year, paper, and they have a volcano plot in that paper that shows the kind of false discovery rate of ASD associated de novo mutations, and the kind of strength of association of de novo mutations in each gene and the resultant ASD phenotypes. And so specifically, SCN2A was probably one of the top genes associated with ASD within that paper. Now, that particular paper did not look at X-linked gene mutations, and mostly because there's such a gender skew effect. And so it was rather difficult to look at gene mutations there for the purposes of their paper. But there is strong literature outside of that paper that looks at, especially FMR1 has been extremely associated with ASD-ID. It's incredibly strongly linked. As I think I mentioned previously, the FMR1 is the Fragile X in particular is the single gene condition that is the most strongly associated with ASD above all other disorders. And then CDKL5, it's much more rare. It is also X-linked, but up to 50% of CDKL5 patients have ASD. And so it is mostly drawn from human data of the penetrance of ASD.
Wonderful. Thank you. The second question was asking if you could speak to the the timeline for LTP versus spatial learning. So the question is, well, for LTP, as you show in your electrophysiology experiments, it can be induced in minutes and lasts for minutes to however long. But the spatial learning that you had took place over days. So how do those two correlate with each other?
So in general, long-term potentiation does last on the same time scale as behavior. It is something that can last hours to days. The only real reason that we didn't record that long is because, again, we're limited by the slice health, the setup that we have, in that recording a single slice for days at a time is just really not technically feasible. And so as a result, even though, again, we could record these slices for 6 hours and presumably show this sustained potentiated response. And again, there have been some in vivo studies actually that have looked at that same thing, the correlation between potentiation and behavior. And so, yeah, LTP lasts on the scale of days. Don't get me wrong. It's just that in terms of the experimental recordings that we do, we only record for up to an hour or two, just to show that the LTP maintenance is stable and continuing.
Right. I know there's also a conundrum where a lot of the scientists that we've worked with and advised here at Precisionary with electrophysiology, the slices, at 6 hours, they deteriorate. And so you kind of question, like, well, if you don't see LTP, then is it because of slice health issues or because you can't induce it anymore, et cetera?
Right. Exactly.
One question got actually the person one of our attendees said that you already answered it. So the last one for you is to ask you just talk a little bit about disinhibition or inhibitory circuitry and the perihinal cortex. And its effect on spatial learning. And actually, you just mentioned that often ASD co-presents with epilepsy. So clearly, there's some sort of role for inhibitory neurons and circuits. So how does that affect your understanding of spatial learning and any project potential?
Right. So that's actually a great question because one of the interesting things about the perirhinal cortex in particular is that it has this very strong innate tonic inhibition. And so part of the literature that's been looking into how the perirhinal may be communicating and coordinating spatial information processing of other regions, including the hippocampus, is that this strong tonic inhibition may act to only refine the elements of the spatial scene and the environment that are salient. And so as a result, it may be that an increase in activity of the perirhinal or long-term potentiation in the perirhinal is going to serve as a spatial salience signal. And so it could be, given the association of ASD and EI balance, it could be that in these mice, the EI balance at baseline is skewed toward too much inhibition in this region. And as a result, even salient spatial cues can't kind of get over this wall of inhibition to create this spatial cue to the hippocampus that this is a salient spatial signal. We need to pay attention to it. And so, yeah, there's definitely a ton of potential for inhibitory circuits, not just in the perirhinal, but across the cortex to be playing a role here. It's just that the reason we focused on excitation as opposed to inhibition is because we do see that, drastically lowered CFOS in the SCN2A het mice. However, it's entirely possible, for example, FMR1 mice and CDKL5 mice, given that they have this association with epilepsy, they may also show hyperexcited circuits. So yeah, there's definitely a ton of research to do, but I do think that part of the reason that the perirhinal may have this sort of such a key role is because it has that kind of high levels of tonic inhibition.
Very cool. Very cool. Well, Dr. Keith, thank you so much for being with us. We are really honored to have you, and we wish the best of luck on your publications and research.
Great. Thank you. Thank you all
Thank
for having
you.
me.